Hi, my name is Kristen Brennand, and I'm a new faculty member in the Departments of Psychiatry and Genetics. I want to thank the organizers for including me in this retreat, and I want to thank my team, not just for joining this retreat, but for moving with me from Mount Sinai to Yale. It will take a few months, but we should all be here by the start of the new year. So, why should a geneticist study brain disease? Well, if this feels like a sales pitch, it is. We are recruiting, so all of you first-year grad students and soon-to-be postdocs, I hope that you realize that brain diseases are common, severe, highly heritable, and complex, arising from the interactions of hundreds of variants of small and large effect. This is the most recent published genome-wide association study for schizophrenia. There are already more than 145 loci that are genome-wide significant. Each of these loci have proximal targets, distal targets, and some of them show context-dependent effects, which I think are one of the things that our lab is most excited about. So how are risk variants themselves influenced? Well, Christina is looking at the impact of donor on risk variants. Michael and Sam are looking at the impact of cell type on risk variant effects. We have open projects on the impact of sex. And Karina and Kayla are looking at the impact of the environment, particularly stress, on risk variant effects. Some of this work extends from previous work looking at a single risk SNP, furin RS4702, that was led by Nadine Shroud. This SNP, at this locus, we think there's one putative causal SNP, as the same SNP is the most significant SNP in the GWAS, as well as the brain EQTL analysis for furin expression. It took Nadine a couple of years, but she applied CRISPR-based methods to achieve perfect conversion of AA genotype to GG. And she was able to show that this single SNP and the non-coding UTR of the furin gene is sufficient to decrease expression of furin, that this effect is microRNA dependent, and that it results in changes to neurite outgrowth as well as neuronal activity. This is where Christina arrived. And Christina is looking at the impact of donor background on this variant effect. And so she's obtained iPS cells from donors with particularly high and particularly low polygenic risk scores for schizophrenia, and she's repeated this CRISPR engineering in these high and low PRS lines. Results are quite preliminary, but it's already beginning to look like we see more robust risk variant effects in the high donor lines, high donor risk lines. And she's beginning to explore this across cell type as well. A second project is also looking at the impact of cell type on common variant genotype effect. In work formerly led by Prashant Rajarian, an MD PhD student in the lab, he was able to look at the 3D genome architecture of neurons and ask how schizophrenia risk variants are linked to more distal target genes, identifying here a particular chromatin loop linking 4 schizophrenia risk SNPs to the protocoherent cluster about 100 kilobase pairs away. Moreover, he was able to show through CRISPR-based methods that removal of these risk SNPs was sufficient to decrease expression of the protocoherent genes. In follow-up, led by Sam Powell, who will be arriving at Yale October 1, we're looking at the impact of these common variant across dopaminergic, GABAergic, and glutamatergic neurons. So he's comparing the 3D genome architecture by Hi-C and Capture-C based methods, looking at relationships between 3D genome structure and gene expression, with the ultimate goal of functionally validating these chromatin loops by creating them in specific subtypes of neurons and looking at the impact on gene expression and neural function. Finally, Michael Fernando is looking at the impact of cell type on rare variant effects. It's been known for years that non-recurrent heterozygous deletions of Nerexin-1 are highly significantly associated with schizophrenia and autism. Nerexin-1 is highly alternatively spliced, with isoform repertoires that vary by brain region and by cell type. Aaron Flaherty, a former PhD student in the lab, together with Shuzhen Xu, conducted the first cataloging and quantifying of human Nerexin-1 isoforms in human brain samples and human stem cell-derived neurons. They were able to show by comparing IPs generated from patients with 3' deletions in the Nerexin-1 gene that there was mass dysregulation of the Nerexin-1 isoform in patient neurons. 50% of the wild-type isoforms were decreased in cases, and an additional third of isoforms were not detected. More unexpectedly, they also identified robust expression of 31 unique mutant isoforms in patient-derived neurons. These mutant isoforms were translated, and overexpression of mutant isoforms in control neurons was significant was sufficient to significantly impair neural activity. This is where Michael showed up, and now he's looking at subtype-specific effects, comparing the impact of patient-specific neurexin-1 deletions in glutamatergic and GABAergic neurons, and the impact of manipulating the wild-type and mutant isoform repertoires in specific cell types and across circuits. Finally, the last question is, how do risk variants sum? Maylin is looking at the convergence of rare and common variants associated with schizophrenia and autism. Itchman's looking at the impact of manipulating networks of genes associated with Alzheimer's progression in microglia and neurons. And Michael's looking at the additive effects of manipulating combinations of risk genes that share or cross biological pathways. And why are we doing this? Well, this also follows up on the unexpected synergistic effects that Nadine observed between common variants. She manipulated 4 schizophrenia risk genes alone, and then from this RNA-seq, generated an expected additive model of what she thought might happen if we manipulate them in combination. She compared the additive model to the empirical results of combinatorial perturbation. The model was quite good, predicting gene expression changes 82% of the time. However, 70% of the time, genes are more down than expected. And those genes that were more down were enriched for neurotransmitter release. Furthermore, some genes were more up than expected, and these 11% of genes were enriched for the rare and common variants associated with schizophrenia risk. And as I close, I do again want to point out that we have many open-end funded projects. They're all co-mentor projects with some of our favorite collaborators. And with that, I'm happy to take any questions. Thank you.